And good afternoon, food friends. So, let's see. The exams have been graded. Hopefully you got the grades. We'll figure out how the posting goes. We were supposed to have an exam this week. I'm behind. I don't want to talk about it. Fine. We're going to keep on going. We'll have an exam next week on some of the stuff we've been talking about, metabolism things, but we'll cover it. Don't worry about it. We're not going to leave you hanging too hard. All right. So, when we last gathered, we were talking about enzymes and metabolism and a couple of specific rules concerning the thermodynamics and thinking about the breakdown of foodstuffs. How do we generate energy? And today, we're actually going to talk about some moderately serious biochemistry, but we're going to keep it simple. We're going to keep it condensed, and hopefully you'll be able to get some stuff out of it. Probably also this week I'm going to post a worksheet that'll help. That if you can do all this, you're good for the exam. It's just sort of a list of things you need to remember. All right. So last time we were talking about enzymes and how they can catalyze reactions, and they can do so at temperatures that are around body temperature and at speeds that are fast enough to actually make things work. And what we can do then is think about these systems in terms of this overall scheme of anabolic versus catabolic. We're going to talk primarily about catabolic type reactions. That is, reactions that are breaking things down and thinking about how we can take a molecule like glucose and break it apart and extract energy from it. So this is a series of biochemical pathways, and I kind of think of it as a grand musical theater, opera, play, whatever. And before we get into it, we need to have our cast of characters, shall we? So just to set the stage here, what we're talking about is oxidizing glucose. Now, last week we oxidized that sheet of paper, whoosh, really fast. That's rapid oxidation. This is going to be a slow, stepwise oxidation. And we're going to do it in a bunch of different steps, and we're going to use oxygen, but it comes in at the end. It's one thing, when you see oxygen hit the stage, you know the show is almost over. So the overall story is this. We're going to take glucose, oxidize it. Oxidation is a loss of electrons. And take the energy and store it. Now, when we set fire to something, that energy was released, whoosh, as heat. That's not going to do us so good. Heat is useful, but not to that extent. We need to store it in a way that will actually work for us. And so here are a couple of the characters that are really critical. And speaking of energy, the first character in this story is this guy, ATP. This is adenosine triphosphate. Now, when I said phosphate, what did you think? Great big negative charge. Right. Because remember, we talked about how phosphates can trigger enzyme action. This really is our energy system, is that phosphate. Now, adenosine triphosphate exists in two forms. It can either be in the triphosphate form, and we write it as ATP, or it can be in the diphosphate form, and we write that as ADP. And what happens is the other phosphate is getting attached to a protein and doing something. So we've got ATP and ADP. And this molecule cycles back and forth between these two forms. Just as a little point, or a thing to think about. This whole thing is ultimate recycling. What we're putting into the system is glucose and oxygen, and everything else is simply being recycled from one form, one state, or another. You're either in the ATP state or you're in the ADP state. Okay, so this is no unsugared sparkling water, flavored, good stuff. I love the bubbles. So this is one of our characters, ATP, and it cycles between the ADP form and the ATP form. So we typically would say ATP goes to ADP, or ADP is rephosphorylated back to ATP. So that's one of our key players. Another key player is our set of electron acceptors. So if this is a Shakespearean play, these would be the two sort of chumps that are always jumping around. They are the electron acceptor twins, and they are derivatives of B vitamins, of niacin and flavines, and these are some different vitamins and things. Anyway, the two electron carriers we want to speak to today are a molecule known as nicotinamide adenine dinucleotide. But you can just call him NAD. Now, please don't refer to him as NAD. That's just rude. I can't deal with that. NAD is an electron acceptor. And when it's in the oxidized state, it hasn't picked up an electron. It's written as NAD. So we'll under here say this is oxidized, meaning it's ready to pick up an electron. Now, if we add an electron to it, remember, reduction is a gain of electrons. If we reduce this guy, we write it as NADH. So that's the form of it that is in the reduced state. And we know when we see it written as NADH, it's picked up an electron and needs to dump it so we can pick back up another one. It also just cycles between these two states. It is either NADH when it's in its reduced form, and it donates an electrons to somebody, and it comes back to NAD. It picks up an electron, and it is now NADH. So we can put a little plus electron right there. All right, so that is one of our key electron acceptors, electron carriers. That's the term, electron carrier. Now, another electron carrier that we're going to see, not quite as popular as NAD, but still, we got to put him on the playbill because he's there. In fact, let's just go ahead and put up here NAD. The next guy is a molecule called flavine adenine dinucleotide. And he is abbreviated, of course, as FAD. And if we give him an electron, we write it as FADH2. So FAD is the oxidized form. FADH2 is the reduced form. All right. So just to have our cast of characters here, we've got FAD as our other character, and then we got a couple of other key players here for sure, and they are trying a new pen here. Our 6-carbon sugar, glucose, And just remember, 6 carbons. And oxygen, O2. So, this is the cast. So I guess what? 4 cast members, 5 cast members? Fine. Now remember, glucose and oxygen, this is the stuff we are going to put into the system. And then these guys are just there. And we're cycling back and forth between these different states. So you go between, you're either an ATP or you're an ADP. You're an NAD or you're an NADH. You're FAD or you're an FADH2. But these 2 components are the only thing that is actually going into the system and being used. All this stuff is simply recycling. And just as a little, sort of an interesting side note, when we think about ATP, we are going to be synthesizing, or I have to think of it as rephosphorylating, ADP to ATP. Each molecule is rephosphorylated approximately 500 times every day. And if you do the calculation, it ends up being about half your body weight of ATP that we are regenerating from ADP every day. Now, of course, your body weight doesn't go up from 150 to 225. It is just staying where it is because you're not changing the actual in number of ATP per cell. You're simply cycling back and forth between either the ADP form or the ATP form. The ultimate in recycling. And we'll see as we get into this a bit. NAD actually can be sold, or is being sold as a supplement. Stupid. Waste of time, complete waste of money. Your cells have a finite, useful supply of these different components, and you're simply cycling back and forth between them. Adding more of it isn't going to affect things because you cycle back and forth at the rate you do, and that's all there is. So, the other little tidbit to keep in mind, sort of thing that we think about, is we're going to be starting to put up our ATP tote board, looking at how much ATP we're generating. And it's this point. We can say that 1 glucose with oxygen is equivalent to 36 ATP. Now, as I said, we're not synthesizing ATP. It really is 36 rephosphorylations of ADP. All right? So every time you're carrying out these anabolic type reactions, or you're using energy up in the form of contracting muscles, you're taking ATP, popping the phosphate off, doing some work with that phosphate negative charge, and you're generating ADP. So if for whatever reason all of your nucleotide is in the form of triphosphate, you ain't got to eat. You probably also aren't doing anything, but you don't have to eat. If all of your nucleotide is in the form of diphosphate, you're in pretty bad shape because you can't carry out any kind of energy requiring reactions. So you're going to be a limp noodle. You have to have these different states. All right. So we can break the pathways or the breakdown of glucose into, I like to do it into 3 different chunks. And we're going to kind of talk about that a little bit. I'm going to give you an outline here, and they do a intermediate step as they break it into 4 chunks. Let's not be pedantic about this for all love. So what we're going to do is start with the first part of the pathway and work through what goes in and what comes out, and then kind of make a condensed Cliff Notes version of that. And then we're going to go into the second part, and we'll probably stop that for today's lecture, and then later this week continue back on with it. But here we go. So the breakdown of glucose starts at the beginning. And just keep in mind, we're owed 36 ATP. Another thing to keep in mind is, where's oxygen in the game? Because, if oxygen is present, if you see oxygen enter into the stage, that means the story's almost done. And second, where is the carbon? Where are the carbons going? So, here is how we start. We're basically going to be setting glucose on metabolic fire. And we start at the beginning with a pathway known as glycolysis. Now, glycolysis was discovered back probably in the late 1800s. It's a fairly well-established pathway. Glycolysis evolutionarily is phenomenally old. Pretty much every living organism on the planet carries out glycolysis in this exact way. So, if you were to go into a bar and sit down next to a bacterium, and you maybe don't have a whole lot to talk to that bacteria about, how's the weather? Yeah, whatever. One thing you can talk about is glycolysis, because that bacteria and you carry out glycolysis in the exact same way, and they're going to go, oh, honey, don't I know it? All those NADs, and I'm not sure what to do with the 3 carbons, and why do we do it this way? Bacteria and you have something to talk about. Here is glycolysis. It starts out like this. We've got glucose, a 6-carbon molecule. And the first thing we do in glycolysis, and frankly, this is kind of backwards because we're trying to generate ATP, we're going to put the phosphates from 2 ATPs onto this glucose molecule. So we're going to go ahead and dephosphorylate, but burn up 2 ATP are being broken down into 2 ADP right off the bat. And that glucose 6-carbon molecule then splits into 2 3-carbon molecules. Phosphoglyceraldehyde. Not that you care, but there you go. Now, these 2 3-carbon molecules are going to go through the exact same pathway. And sometimes the books will try and superimpose one over the other as ways of saying, okay, this is happening twice. But just remember, it's happening twice. So, the 3-carbon molecule is now actually going to dump an electron. It's going to finally get oxidized a little bit. And if something gets oxidized, somebody else has to get reduced. We need an electron acceptor. Here comes our friend NAD backing up to pick up an electron. Beep, beep, beep. And it becomes NADH. It has been reduced to NADH. And all the carbons are still there. We have a 3-carbon molecule, it's a different 3-carbon molecule. Now, finally, we're going to actually extract some energy in the form of ATP here. We're going to use the energy in these chemical bonds to add phosphates. So we now, with each 3-carbon, have the energy to take 2 ADP and convert them to 2 ATP. And the same thing over here. 2 ADP are converted into 2 ATP. And just as a little note here, those of you that maybe lift weights, you perhaps have heard of creatine as a supplement. There's an enzyme called creatine phosphokinase that is peripherally involved in this regeneration of ATP here. And so when you're taking creatine, the idea is you're kind of hopefully sort of biasing this part of the reaction. And There's vague evidence that it has some effect positively, but not a whole lot of effect. And I don't think that taking creatine supplements are going to do you that much good. People say it causes you to gain water weight, very small amounts. Fairly sketchy evidence for it. I ain't buying it. There's a couple of papers about it, but it's not real good evidence. So there you go. Now, at the end of glycolysis, we're left with two molecules of the 3-carbon molecule called pyruvic acid, or pyruvate. It's a 3-carbon molecule. So let's just kind of write up here a very condensed version of what's going on here. Really, that is what you actually need to know. So write this down on a little post-it note, memorize it, and you're good. We can say this. Glycolysis, what goes in is the 6-carbon glucose. What goes out is 2 3-carbon pyruvates. So what we can say is you didn't lose no carbon yet. All right. Now what also goes in is 2 ATP. What comes out is 4 ATP, 2 from each one of these guys. And another thing that goes in is NAD. In fact, we need 2. So 2 NAD, and what comes out is 2 NADH. All right, now you might say, what about the extra ADP required to make these 4? Yeah, I don't really count that. It is assumed that you've got this big pool of ADP. Otherwise, you wouldn't have to be eating. And so ADP is always floating around. You're lucky if you don't have any ADP floating around. That means that you are totally tanked up and ready to go do something. Now go off and do something so you can have more ADP around, but there you go. Kind of as a good toast. May all your nucleotides be in the triphosphate form. All right, so that is our tote board, and that's really all you need to know is what goes in, glucose, ATP, NAD. What comes out, pyruvate, the 4 ATP, 2 NADH. Now, I ain't no accountant, but what we can say is, as far as our tote board goes, is this. We have a net gain of 2 ATP. Now, I erased it, but you remember what I said was 1 glucose gives us 36 ATP. We have generated 2. There are 34 still to go. Let's see where they go. All right. So the next step is this. Eep, 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 eep. The next step is this, and this is where you can kind of lump and split, is that this is known as oxidation of pyruvate. Pyruvate, one at a time, gets escorted into a special spot in the cell called the mitochondria. We'll have more to say about the mitochondria in a bit. Just know that the mitochondria, it's what's known as an organelle. Glycolysis takes place in the cytoplasm. This next step is taking place in the mitochondria. As the pyruvate is going into the mitochondria, a carbon is kicked off as CO2. And because the second one's following right along behind it, another CO2 comes off. We're going to just follow 1 pyruvate for now. And this means that we're going to have now a 2-carbon molecule coming into the mitochondria. And just not to be bothered by it, but we also, whenever you kick a CO2 off, just remember that that's liberating an electron. And so we also have an NAD being reduced to NADH. Okay? So, the mitochondria is what's known as an organelle. And we'll get into the structure of the mitochondria next time we talk. But for now, if the cell is the size of a beach ball, the mitochondria is the size of, one of those kind of squishy circus peanut things. Or if the cell is the size of a pumpkin, a mitochondria would be the size of a jelly belly. Fine. One cell might have a dozen mitochondria. Some cells, like muscle cells, might even have 100 or more mitochondria. So you can have a lot of them. All right. So the pyruvate makes its way into the mitochondria and it loses a carbon. Now, remember, two carbon molecules, they're known as the ethyl group, but they're also known as the acetyl group. This is a molecule that is called acetyl, and it gets complexed up for just a little brief moment with a sort of a vitamin guy that's called coenzyme A. And that means we refer to this two-carbon molecule as acetyl-CoA. The only reason I say this is, and frankly, the biochemical reason why it gets associated with CoA is probably very subtle, and I don't know what it is. But that's the name of the molecule that enters into the next cycle, or the next system, known as the Krebs cycle. Named after Hans Krebs, a German scientist that left Germany for England, I think right after World War I, and developed this system. And it's actually a rather clever kind of system going on, just for those of you that are true nerds. Glycolysis is a linear thing of where you're going from a higher energy to lower energy. The Krebs cycle actually is a cyclic thing. It makes it a little bit more complicated, but it's actually a very clever mechanism that goes on. It works like this. We've got a 4-carbon molecule that is an acceptor for the 2-carbon molecule. It comes in, the acetyl-CoA goes in here, and we now have a 6-carbon molecule. This, by the way, is citric acid, one of my favorite acids. And in fact, sweet tarts and all good sour things come from the components of the Krebs cycle. So folks at ADM are taking yeast and different microorganisms, cracking them apart, and taking apart or extracting some of the parts of the Krebs cycle to sell for real sour candy and for Kool-Aid and for sweet tarts. So of all the cycles around, this is going to be my favorite because I love citric acid. This 6-carbon molecule comes around. We're going to lose a carbon as CO2, and we have now a 5-carbon molecule. This molecule loses a carbon as CO2, and we have a 4-carbon molecule. And this 4-carbon molecule rearranges itself a couple of times, and you're back up to the top of the cycle. It picks up another 2 carbons and keeps going. So each one of these pyruvates makes its way through here. Now let's just take a little moment. Pyruvate's 3-carbon. That's carbon 1. That's carbon 2. There's carbon 3. Okay. Once you pass through this cycle, there ain't no more carbon. I mean, the next one comes through and it dumps its carbon. This is where the carbon dioxide that you exhale out is coming from. So, you break down glucose into carbon dioxide, and you've done it right here. Now, there are a number of metabolites and things that are happening here. Some of our cast of characters are featured in this little song and dance number. Let's go through here. We got an NAD going to NADH here. The same thing is happening here and here. I generally don't count them. I usually just say it's a buttload of NADH, but a buttload is not a very accurate term. We also have an FAD becoming an FADH2. And we've got a molecule of a diphosphate being phosphorylated to become a triphosphate. Okay, this is where it starts to get really tedious. It's not an actual ATP. It's actually a guanosine triphosphate called GTP. It gets put into the mix as an ATP. Do we need to worry about this? God, no. Let's don't worry about it. How about we just call this a nucleotide triphosphate? It's generated. We have an NDP being rephosphorylated to an NTP. If that's too confusing, just say ADP goes to ATP, but it's not completely accurate. I don't care. Fine. Your answer would be cool either way. Like if you were to see a multiple choice question, it'll probably be ATP. Leave it. Don't get picky on me. All right. So that's the cycle. And now what we can do is write our what goes in, what comes out list again. So for the Krebs cycle, we've got N is the 3-carbon pyruvate, and let's just say lots of NAD and 1 FAD, and what comes out is 3 carbon dioxides, lots of NADH, and an FADH2, and then 1 ATP. We'll just call that guy an ATP. All right. And not bad. Now, here is the interesting question. A, no O2 yet. B, we generated a grand total of 2 more ATP. Whoopee. So we've got, for both the carbon molecules going through, we generate 2 ATPs. We generated 2 ATPs from glycolysis. That means that so far our tote board is glycolysis is 2 ATP, Krebs is 2 ATP. Our total is supposed to be 36 ATP, and I'm not great at math, but I can tell you that somebody needs to call in and light up those lines because we ain't got enough ATP here yet. We only have 4 ATP out of the 36 that I'm owed. Where the hell are the other ATPs? Glad you asked that. We will pick up on that next time we discuss, which will probably be tomorrow or Wednesday. So, hopefully you watch this. Hell, you might turn the damn thing off. Cheers.